So this data was taken from the publication Modeling Precision Treatment of Breast Cancer, which was featured in Genome Biology in 2013 and produced by researchers at a company called Genentech. The publication is interesting because the authors assembled a dataset of about 50 different breast cancer cell lines that represent the whole spectrum of breast cancer variations. The cell lines have genomic, transcriptomic, and proteomic data, as well as an efficacy measurement of commonly used and experimental cancer drugs. If we look in the paper abstract, we can find relevant terms to this project: molecular profiles, selecting features specific to each subtype of patients, and a use case for various machine learning methods to find answers about efficacy of drug treatments for the right patients. This brings me to this flowchart that places our analysis into perspective. When we analyze data, we want to answer a question. The question leads into the dataset, which has to be prepared. Before we even get the analysis step or answer any of the questions, we need to know what data we are dealing with, what are its properties, and how does it describe the conditions that we are interested in. In the next few lessons, you will learn about this type of data and the way we can explore such data using data visualization, statistical analysis, and machine learning in Python. Ultimately, you will see how analysis of transcriptomic, genomic, or metagenomic data can lead to important answers posed in this publication and many other similar projects.